So last video we left off with our pre-mRNA molecule filled with a bunch of RNA nucleotides, A's and U's and C's and G's. It's this long string, right? One single strand of RNA. This is, again, I called it pre-mRNA because we're not done modifying it for it to be ready to be translated into protein. First, we got to have some mature mRNA. So I have a picture here, and we need to talk about some of these processes, which we are in which we are going to modify this mRNA for two reasons. Reason one, a lot of this is junk. Some of this needs to be pulled out so we can put together a functional protein. Another reason is RNA doesn't survive very long outside of the nucleus because you have a bunch of enzymes that are dedicated to breaking down mRNA. So we got to protect it for some amount of time. The first thing we got to do is we have to, as I said initially, is we have to remove something called introns. Introns. These are the regions of the mRNA that are effectively junk. They aren't used, right? These little gray areas right here are called introns. So non-translated regions to be pulled out. That is what an intron is. An exon, this is a translated region. Translated region, right? So we should be left after we remove our introns with only exons. And the way it works is, and this is just kind of, you don't necessarily need to know this, but it's nice to know. You have these little things called SNRMs, pronounced SNRPs. SNRPs. These are small nuclear ribonucleoproteins. Small nuclear ribonuclear proteins, or SNRPs. These basically come in, and this protein complex causes a bend here in this intron and causes it to essentially pinch off in this process in which we're getting rid of this junk, right? These introns that we're not going to use. We're going to talk a little bit later about why sometimes what's in these introns isn't necessarily junk. All right, the second part of processing that needs to be done is we need to prepare for cytoplasm, right? We got to prepare them for the cytoplasm. In the cytoplasm, it's not like the nucleus. There's a bunch of enzymes. Enzymes that attack RNA, right? And they break down RNA, no matter if it's yours or not. It doesn't matter. And the reason for that is because you could have RNA molecules injected into your cell via a virus or something like that, in which you have foreign RNA that needs to be broken down so it doesn't get translated, and you have some malicious proteins in there, right? So in order to protect this mRNA for some amount of time, we basically add a cap on one end called a 5' GTP cap. And on the other end, we add a poly A tail. Now, poly, as you know, means many. A refers to adenine, right? So essentially what this is at the end of this protein here is just a bunch of A's, a bunch of adenines, right? Long tail of them, anywhere from 50 to around 250 of these things. The more A's you have on the end, the longer lasting this mRNA is going to be, because your enzymes, which are coming through like Pac-Man and eating this mRNA, it takes them longer to get down here, right? As they eat from this side over here, this vulnerable side, it takes them longer to eat all the way up into the relevant parts of this protein if it's a longer tail, right? This GTP cap it kind of gives it a cap on the end over here. There's basically a 3-phosphate link to a guanidine up here. And that caps off this mRNA. Now, once you have the introns removed, you have your 5' GTP cap, and you have your 3' poly A tail down here at the other end. Now, your mRNA is fully mature, right? So it's ready. You should have a long tail, poly A tail here, right? Bunch of A's, again, about 50 to 250, right? Somewhere in that range typically. And you have your 5' GTP cap up here at the other end. And you have your coding region. Again, introns are removed. Introns have been removed by this point. And your mRNA is fully mature. Now it's ready for the cytoplasm, and it's ready to leave the nucleus and enter that dangerous area, right, where it's gonna eventually be destroyed. But before it's destroyed, it's going to be translated possibly many times. One thing I didn't want to talk about is sometimes, if you'll notice here, DNA to RNA, you'll see these exons over here, right? And I want you to pause real quick and try to remember, are exons the region of mRNA that code, or are they the junk regions of mRNA? All right, should have said that these are the regions that code. The rest of this here, these are introns, right? However, sometimes you can cut out portions of different exons to create different transcripts, which leads to a different protein. Now, what do I mean by that? Look at this first mRNA molecule over here. You have this blue exon 1, you have exon 2, exon 3, exon 4, exon 5. So you have all of these exons, and they get translated into protein A. Well, you might have a different splice of this mRNA, right? That those SNRPs might come in and snip out, in this case, that exon 3. You'll notice you're missing exon 3, 1, 2, 4, and 5 only, right? So you've essentially treated this exon here as an intron for the purposes of this splice, and you end up with a different protein. Same for this translation over here. Which one are we missing? Oh, we're missing exon 4 right here, right? So that's going to give you yet another different version of a protein, right? So this is kind of the why it's kind of nice to have these introns removed, because you can create these different splices, which leads to different proteins, all for one gene. And that is going to be it for transcription RNA processing. I do want to review where some of this stuff takes place. Let me change my color here. Again, we look at our cell here. All that we've been talking about so far, DNA to mRNA, takes place here in the nucleus. This is where the copy is made, or where transcription takes place. Of course, now we have our mRNA leaving, right? It's ready to leave now. It's got a GTP cap up here, GTP cap on this side. It's got a poly A tail with a bunch of adenines over here, right? And it's ready to essentially leave and go over to a ribosome somewhere in the cell, either here or over at the ref ER. But the point is, it's ready to leave that nuclear pore and brave the cytoplasm here, right? So transcription, first process we talked about, we're done with that, and it takes place in the nucleus. The rest of this portion right here, this translation portion for eukaryotic organisms, is going to take place in the cytoplasm. And that's what our next videos are going to be about. So if you need to, make sure that all your drawings are good. Make sure you've listened carefully and you have some good stuff written down. Maybe even some of the stuff I didn't physically write down on the slide. And you should be good on transcription. Translation is up next. All right, thanks for listening.